All right, so biodiversity. So biodiversity revolves around kind of ecology and number of species and what communities can hold what number of species and how those species interact. And you can start comparing things like how abundant something is versus if we're talking about a community, for example. Remember, a community is a group of organisms that interact, right? Like this, montane or kind of prairie here that has a ton of flowers in it, right? So we could call this a community. So we got not only the number of different species, which is called biodiversity or diversity richness, but also the abundance of species. Some might be more abundant than others, or more apparent than others. And then there's obviously interactions between these that affect their abundance and species richness. And those start leading into our discussions on ecology. So basically, once we finish this lecture, we are going to move into ecological principles and start applying some of these things that we've talked about as far as evolution and biodiversity go. We will also do a taxonomic review of life on Earth here through this lecture. So the first thing we want to do is just make sure we know what biodiversity is, which is the number of species occupied in some space, right? That could be a really small local space, like we might be interested in a local garden, right, or forest, or it could be Africa. What's the biodiversity of Africa? How bio-rich, or how many species do we find on the continent? So it could be very global, all right, or very large, or it can be very small, it can be very local. But what biodiversity is the measure of species richness, and the evenness of the distribution of those species across the community or across a certain area. We can measure it in many different ways. I mean, you can simply count numbers, right? There are 30 species in this garden. There are 250 species in this garden, right? Or forest or mountain range or on this continent. We could also do abundance. How many of each particular species we have, and then do a comparison between species richness and abundance. We can also look at how even we distribute the numbers across the different species that you see in a community. So there's a whole bunch of different ways to measure this, and I'm just going to give you a few. You'll get introduced to this a little more in ecology in our later lectures, and then obviously doing some of it in practice when you take ecology with Dr. Sweets. And then we want to know what type of biodiversity exists in our world. We have great biodiversity, but it's concentrated in certain areas. Here's one great example, coral reefs. Most marine fish are found on coral reefs, somewhere between anywhere from 30 to, I've seen 60, 65% of fish life live on coral reefs, which make up 0.01% of the entire ocean. So a very small area has most of the biodiversity. Same thing happens with animal biodiversity, as you'll see. So how many species exist worldwide? Heck, we have no idea, right? If we're talking about all life, how many species exist? We have only described about 2 million, roughly 2 million. It's actually more than that now. So I made this slide several years ago. So we're up in the 2.5, close to the 3 million range now, maybe, if you were to actually go and look at the most recent literature. The actual number is unknown and is estimated by conservatives to be about 1 billion species or less, or over or around 6 billion species. Almost all are going to be bacteria, all right, or prokaryotic. Bacteria are by far the most numerous organism on the planet. I'm talking about sheer numbers. They're found everywhere. We call that cosmopolitan when I say it's found everywhere. And they're probably the most species rich. The biggest problem is describing individual species or types of bacteria. Of course, the animal we know most about are animals, right? I mean, these are the ones most of these 2 million described species are actual animals. 1 million of those, or half of that number, are insects and spiders and mites. There are, arthropods is the group name you'll learn, or the taxonomy name you'll learn. We have about 1 million or more insects that have been described All other animals are around the 400,000, 450,000 mark as far as individual species names. Plants, pretty similar, most of those being terrestrial plants, plants that live on land. Then you got a little sliver here of algae, little sliver of fungi and protists. Here's bacteria in similar forms like archaeans, and then a really small sliver of viruses. There are actually several, several thousand viruses that have been described across our planet. So this kind of gives you, predicted, I should say, described species or so, and probably what the predictions are. New species are being added to this list every day. Everywhere, we call that cosmopolitan when I say it's found everywhere. And they're probably the most species rich. The biggest problem is describing individual species or types of bacteria. Of course, the animal we know most about are animals, right? I mean, these are the ones most of these 2 million described species are actual animals. 1 million of those, or half of that number, are insects and spiders and mites. There are, arthropods is the group name you'll learn, or the taxonomy name you'll learn. We have about 1 million or more insects that have been described. All other animals are around the 400,000, 450,000 mark as far as individual species names. Plants, pretty similar, most of those being terrestrial plants, plants that live on land. Then you got a little sliver here of algae, little sliver of fungi and protists. Here's bacteria in similar forms like archaeans, and then a really small sliver of viruses. There are actually several, several thousand viruses that have been described across our planet. So this kind of gives you, predicted, I should say, described species or so, and probably what the predictions are. New species are being added to this list every day. So this is our current understanding of diversity. This is probably the real picture, right? Is there are going to be a lot more prokaryotes than anything else out there. We just have a really hard time with the technology we have today describing individual species of bacteria. All right, and you might ask why, right? Well, why is that? Well, think about bacteria. They're small and simple. They all really look alike. I mean, there's not very much you can do with such a simple game plan, if you will, or body plan. I mean, they're a cell wall, a cell membrane, DNA, and ribosomes, right? And some cytoplasm. They do have some variation in shape. There are 3 major shapes of bacteria. Many of them grow on petri dishes kind of differently from one another. And some of them stain differently than one another. But the biggest problem is that bacteria defy our species definition, right? We define species in a number of different ways. I've given you a number of ways. But the one we like the best is biological, right? Which means they have to reproduce sexually. And bacteria and archaeans don't reproduce sexually. They're asexual. They're mostly mitotic. They also can pass DNA around like they're lollipops, if you will, right? So they can do horizontal gene transfer, and that really makes their genetics very complicated. So they're very genetically diverse. They're very metabolically diverse. They're very similar in what they look like. It's kind of hard to tell who is who. The other major problem is we can't see them. They're really small. They're hard to visualize. And most of the bacteria that we want to visualize or study have to grow on a petri dish. And we don't have the ability to do that. A lot of them won't grow on petri dishes. At least they won't grow on what we try to grow them on. You may not, so I'll give you an example. You may not know this. There is a type of agar. You know how you grew yours on your petri dishes? That's called blood agar. It's made out of blood. And it'll only grow certain bacteria. Isn't that weird? So who knows what you could grow bacteria on if you tried. So standard agars that have standard nutrients on them don't grow every bacterium. Remember, a lot of bacteria think oxygen's toxic too. They don't use it. I say think. They don't use oxygen, right? So they're anaerobic and oxygen's toxic. You couldn't even grow them in this room. You would have to grow them in a chamber that has no oxygen in it. And so they are tough to kind of categorize into individual species. As our technology or ways in identifying and describing them increase, we will start to increase the number of species that we see. Bacteria are absolutely everywhere, as you'll see, and they're found everywhere. And there's a good reason for that also, because remember, they were the first organisms to actually arise on the planet, were prokaryotes. So that brings us to kind of the next question: is where are species distributed? So here's one wonderful place. It's one of my. That's my happy place, right? So tropical jungle is a great place. This is a beautiful tropical jungle with a little river going through here. So where are they distributed across the world? They're not evenly distributed. Most of you might know this if you've been to the tropics, if you've been in a rainforest. If you have never been in a rainforest, try eventually someday to reach a rainforest and check it out, because those are what are considered biology or biological or biodiversity hotspots. So most tropical rainforest is the hotspot terrestrially, or a place that's called Mediterranean type climate, which I'll talk about in a few minutes. So these biodiversity hotspots typically have very stable environments with lots of nutrients, lots of water. Temperatures don't change much. Rainfall is very predictable. Typically it could be year-round, or there's a rainy and dry season. And there's very little extinction that happens because the environment's stable if it's undisturbed. Because of that, and because of the primary production, you guys all heard the term primary production before? It's when it's an environment that has a lot of plants or a lot of plant biomass, which can produce a lot of photosynthetic product, right? And when you can get a lot of photosynthesis going in a system, then you have lots of diversity that can happen because you got lots to eat, right? A lot of plants to eat. Also, those plants provide habitat. They provide niches for animals, or ecological roles for animals to occupy. So not only are these biodiversity hotspots heavy in niches, or lots of different types of niches, and have a lot of plant production, a lot of primary production, this often leads to the ability to have very, very specialized animals that are going to be called niche specialists. And we call these endemic species, which means that they're found only there. So for example, I've done a lot of work in Costa Rica. I've taken students to Costa Rica about 10 times, well, 10 times overall in the 2 places that I've taught. That is a major biodiversity hotspot. There are lots of organisms there, lots of rainforest, lots of different types of environments, and there are tons of endemic species that are found just in Costa Rica and found nowhere else. That's what an endemic species is. And the reason for that is because of the ecological diversity there. There are niches there that are very specialized and only found there, which species have learned to occupy or evolved to occupy by natural selection, and they're only going to be found there because that's the only place you can find that environment. These jungles are often isolated by mountain ranges or dry patches or desert or something that allows them to become reproductively isolated from other similar habitats and allow for a lot of natural selection and evolution to happen. Terrestrial species, those that live on land, are typically concentrated in the tropics. Just to give you kind of a number, somewhere around 80% of all living terrestrial species live in the tropics, all right, between the equator and the subtropical lines that keep in that area where it's always warm, it's always wet, there's always 12 hours of daylight, 12 hours of dark time, and it's very stable often with very stable or consistent seasons, typically a rainy and a dry season. So tropical forests, these could be rainforests, they could be deciduous forests. They only make up about 7% of the land mass. That is, they only cover 7% of the land mass, but more than 50 plus number of species, terrestrial species, are found in tropical rainforest. The ocean covers 70% of the earth, and most of it is species poor. There aren't very many species spread across the ocean. Most of them are concentrated in places where there's shallow water, for example. And lots of sunlight again, so you get lots of primary production. Coral reefs being the major hotspot, which only covers about 0.1% of all of the ocean as far as total mass or volume, however you want to think about it. Lakes and rivers also play a large part for fish. About 45% of fish are found in lakes and rivers, and they only make up a really small percentage of the Earth's water, both marine and fresh. And then there are these Mediterranean-type communities that sit in the right place climate-wise on the planet. We'll talk about why and how you get these different types of climates across different latitudinal ranges across Earth when we talk about big biomes and weather patterns. But Mediterranean-type communities are very nice, right? So think of Greece, think of the Mediterranean region. You can find these also in Southwest Australia, in Chile, in South Africa. They look like this. No real big trees, a lot of different shrubs and small trees, a lot of diversity in plant life. So a lot of primary production and diversity leads to lots of niches. So you get large species diversity in these regions. They typically are mild. Their temperatures have mild winters. They can get pretty hot, but they have wet and dry seasons. And it leads to a lot of species richness, both plant and animal species richness. So here's a map of obviously the continents here, and you can see this is where all the terrestrial species are. So you can see all of them. They're really concentrated in the tropics, right, where it's warm and stable. Okay, there are lots of different ways to measure biodiversity. As I already mentioned, you can just simply count number of species, right? I mean, that's the easiest way to do it. But there are other mathematical models. I'll introduce you to just a couple of the more common ones that you see in general biology, and you'll probably practice some of this later on in your ecology class. But we can measure biodiversity using many different models. Here are a couple different types. Again, you're not going to have to do these. I might even show you, I'm going to show you a mathematical formula in a minute. You don't even have to worry about that. Really, I just want you to know definitions and kind of what these do. So species richness is simply the number of species present in any place. All right, and the problem is, okay, that's an interesting generic definition, but you know, ecologists really want to know how big the area is, right? So you want mathematical models that actually measure species richness based on size of the community or locale. So if you want to do very local, very specific, very small, if you will, species richness in a local area, an area that has a small land mass or water volume, then you would do alpha diversity. So for example, if we're in the state of Indiana and we pick a lake and we want to know the biodiversity in that lake, we would use alpha diversity because it's that one lake, right? So alpha diversity is the number of species in a single community or small local area or space. Gamma diversity is the number of species in a larger geographic area. So alpha diversity is that one lake. If I did all the lakes of Indiana, then I would use gamma diversity, right? I'm not now using one community. I'm combining a bunch of communities in a particular region. And I can make this a big region like the United States or North America or Africa, or it can be regional, Midwest, Indiana, would be gamma diversity. Beta diversity links alpha and gamma so you can compare your local to your regional diversity. And it allows you to, kind of compare diversity locally versus what overall diversity is in a region or area, which will house the community that you've came up with your alpha diversity number with. So this is beta diversity is pretty simple. You just divide gamma by delta, gives you a number, right? And so how would you use these? So here's an example, just one example. Lots of different ways to do this. So you got 3 different regions, right? And this allows you to compare the regions next to each other. So the first one up here, we have 3 mountains, right? Our alpha diversity, if I want to know what our alpha diversity for each mountain is, it's about 6 per mountain. And if you look at this, if you count them, if you were to divide all these up by 3, because you got 3 mountains, you would get 6, right? 6 individuals, or around 6 individuals for each mountain. The gamma diversity is only 7. That is the whole region. All 3 mountains combined, there are only 7 species, right? So when you look at these 2 numbers, that simply tells you that each mountain is very similar, isn't it? Right? And if you look at it, A, B, C, D, B, C, D, E, A, B, C, D, right? Most mountains have the same species on them. So the communities here are very similar. The species you find on each mountain are very similar in the region. And there's very little isolation between the populations, right? They're obviously moving from mountain to mountain, right? Or they can disperse. There's no limited dispersion of species. And so you have kind of uniformity across a region. Therefore, your beta number is really small, right? So high alpha number, that is a large number of species within each mountain. Low beta means that the mountains are very similar, right? Or the communities are very similar on these mountains. Region 2 and 3 are much different. Notice your alpha is smaller, your species number is bigger, right, overall, which means that your beta number is more. And what that tells you is there's a lot more diversity in the communities. So you can see A, B, and C are only found on this mountain, right? And that's much different than over here, which is D, G, H, and I, and J, right? So you got species that are localized to certain communities, which means there's probably a lot more diversity in what the community looks like. And there's isolation or selection that's preventing everybody from moving around, right? That's seen even heavier down here in region 3, where there's a lot more isolation and your number goes up. So the higher the beta number, the more different and diverse individual local communities are in a region, and typically that is maintained through isolation or the prevention of dispersion, and typically natural selection, right, or evolution. So this is one way you could start to, compare and look at local versus regional diversity. One of my favorite ways to measure biodiversity is what's called functional biodiversity or functional diversity. This not only takes into account the number of species occupying a community, but their role in that community. All right, and why this becomes important? Because roles or number of species trying to do or perform a role can change biodiversity. All right? Removing a species with a specific role will change biodiversity. So it's, for me, kind of more rich or more interesting, because it allows you first to say, okay, I have this many species, and then look at each species and what its role is or what niche it occupies in that ecosystem. So you're basically taking biodiversity now and applying ecological principles, which is where we're going. And that's what functional diversity is it better addresses not only how many species are there, but what the ecological function of each organism is. So diversity of organism is categorized by its ecological role. So a great example of this, apex predators. Have you heard about apex predators before? A predator that sits on top of the food chain. Great white shark, for example, right? They sit on the top of the food chain. As do tuna, which is, again, another larger fish. So if you have a community that has one apex predator, it'll have a different biodiversity than a community that has 2 apex predators, because they're eating different things, right? Which is going to affect the selection and evolution of the animals below that are being eaten, that are the prey. If you remove an apex predator from a community, it's going to change the biodiversity, right? Because now you're no longer being eaten. And if you're no longer being eaten, then you're eating other things, and the whole dynamics of the food web changes. This is a great example. If you're not eating starfish or sea urchins, they become very abundant, right? And when they become very abundant, they can become very damaging or dangerous to an ecosystem. And we'll talk about examples of this. And of course, if you're snorkeling, starfish look pretty boring, but they're pretty voracious predators. And it might take them forever to find their prey because they move really slowly, but they do eat. And if they do reproduce quite readily. So they can, if not controlled, really change the biodiversity for the worse when we start disrupting ecosystems. So that's where it gets interesting, where you can start applying, oh, this is what diversity looks like in an undisturbed community. Here's what diversity looks like in a disturbed community. Why? And you can start to see that you're probably going to remove some ecological roles from a disturbed community, which is going to affect the overall biodiversity. So those are kind of things where we're going, what we're going to start talking about as far as ecology goes. Make sense? Okay, so how do we measure it? There are lots of different ways to measure it. Don't get scared by the math. Who cares? You're not going to have to do it, right? They would give it to you. You could plug numbers in. It's just math. But everybody starts tuning me out when you see a mathematical formula, right? Here are two ways you can do that. One of them, and these are two very common ways you can measure or look at biodiversity. So the Shannon's diversity index weighs abundance versus evenness, right? And so, what the heck does that mean? Abundance is actually the number of a species. Do you have 100 of them, 1,000 of them, 10,000 of them? 2 of them, right? Evenness is how distributed are species as far as their abundance. So it gives you kind of an indirect measure of richness. So let me give you an example. So if you have a community that has 1,000 individuals in it, that's all the community can hold is 1,000 individuals, right? If you use evenness and you evenly distribute the species across that 1,000, you're going to have a lot more species than if you do it unevenly, right? So, for example, if I say there are 1,000 individuals and I'm going to evenly distribute them across 20 species, right, then you'll have a certain number per 20 species, right? If I say you have 1,000 species and 900 of them are one particular individual or one particular species, then you don't have a lot of room for any others, do you? Does that make sense? So abundance affects resources in a community. That is, you got to remember, resources aren't infinite, right? Every community has a certain resource limit. And the number of individual species and how many species a community can hold based on the number of each individual species is different depending on how many resources you have. So that's what the Shannon Diversity Index kind of does. It kind of weighs this. If you have high Shannon diversity, you have high richness and evenness across your species. It's typically thought to be better. If you have low Shannon diversity, then you got higher abundance of some species and not as many species as you would in other ecosystems. The Jacquard coefficient of community similarity, these are two you'll use in ecology class. These compare two communities. You have a community with this species richness and a community with this species richness. You want to compare them. And how do you do that? There are mathematical models that let you compare them, and this is one example of that, so that you can compare communities and their richness to each other. Whew. All right, we'll get out of that kind of stuff. Now now we're going to start our taxonomic review of organisms. Today we're just going to talk a little bit about bacteria and their ancestors. I obviously told you there's a lot of biodiversity, particularly in these species-rich spots, or these what we call biodiversity hotspots. How did all that diversity originate? You can already tell me that, right? Or at least a little bit of the story. You could make an argument now. You got natural selection, you got catastrophism, you got niche partitioning, you got niche openings, right? You have species diversity, you have punctuated equilibrium, you got gradualism, you got all these processes that have been working on the planet through Earth's history. And we know that life first arose in some chemical evolution, right? Some spattering of chemical evolution. And here's a great example of where maybe that could have happened, which this is a volcanic vent here on the bottom of the ocean. and you're spewing all of these chemicals, which include carbon-based chemicals, some methane, right? Hydrogen sulfide, a number of small molecules that are going to interact in a very energetic environment, this one being heat-driven, can produce the proper macromolecules for life, right? We talked about that. And if life originated out of that, then we're going to get some universal common ancestor, right? And we call that universal common ancestor LUCA, right? Which is the last universal common ancestor. Do we know what LUCA looked like? No. We have no fossils of LUCA. Was LUCA one single organism? Probably not. It was probably a bunch of them, right? Many species, some of them doing better than others, some of them going extinct pretty quickly, all of them probably sharing a lot of genetic information as they developed it because of horizontal gene transfer. And then obviously would reproduce probably pretty similar to binary fission, or be kind of bacteria-like, right? We would think, or very prokaryotic-like. From LUCA, or out of the chaos of the chemical revolution and the early evolution of LUCA, then we get our three major domains that we see today, which are bacteria, which is the oldest domain, and then archaea and eukaryotes. Notice archaeans and eukaryotes are sister to each other, right? They're more closely related to each other than they are bacteria. Do you see that? Right? Bacteria are kind of the base, if you will, or the ancestral group for the rest of life, particularly eukaryotes. So LUCA is the last universal common ancestor. Was probably many different species or many different ancestors. There was a ton of what we would call horizontal gene transfer, or I call it horizontal gene transfer chaos in the beginning with the exchange of genetic material. And then out of all of this, the chemical revolution and this chaos of horizontal gene transfer are your three major domains that we see today. And here's kind of what a tree would look like if we could root it down here somewhere with a bunch of different representatives of common ancestral community, or a bunch of LUCA individual species arise, live, share a lot of genetics, and then eventually what comes out of that, or nested out of that, are your three major groups, right? So obviously first bacteria, and then archaeans, and then between bacteria and archaeans, remember we get endosymbiosis to produce our first organelles. So we got plastids over here and mitochondria, and then the evolution of the eukaryotic line with those new organelles. And so you end up with these three major domains. And of course the one we know best are the larger, more diverse, as far as size and multicellularity, the eukaryotes, which we are part of. What we think LUCA possessed, and again, this is a hypothesis. We don't have any fossils or any, direct evidence of this. But if you look at, you can deduce by looking at bacteria, archaea, and eukaryotes, that LUCA possess synapomorphies that bacteria and archaeans now have, right? So those synapomorphies would separate bacteria and archaeans and LUCA, or at least the common ancestor, from all the rest of primordial life. They have, it looks very bacteria-like, very prokaryotic-like. No nucleus, a nucleoid, no membrane-bound organelles, one chromosome, and many plastids, these little DNA rings that we can transfer genes back and forth to do horizontal gene transfer. Probably a flagellum, something to move, right? Unicellular, small size, cell walls, and they use binary fission to divide or to do their vertical gene transfer, right? And this isn't really reproduction, right, as we know it, like meiosis. So it makes it really hard to define species. But these are probably pretty similar to the traits that these last universal common ancestors had that gave rise to our three major domains that we see today. So we're going to start with bacteria and work our way through the eukaryotes. So we have a slide or two, we have 3 on bacteria, 1 slide on archaeans, and then a bunch of slides on the eukaryotes, which you're probably more interested in, right?
Mm-hmm. If you said like the eukaryotes kind of diverge from like archaeans, and why is it not archaeans and eukaryotes like swapped in that thingy? Because like when it looks at it, it looks like your eukaryotes kind of like diverge first, and archaeans came up.
What do you mean? Why isn't he over here?
No, like why is the archaeans not where the eukaryotes are? Because it looks like the way it looks like is archaeans like.
Well, you can flip these. Oh. So you can rotate any phylogenetic branch. Yeah. Oh. Yeah, you can rotate it.
Because the way I looked at it is it looks like archaeans like diverge from eukaryotes.
No, they're just sister to each other. And actually, we don't know. I mean, archaeans could have diverged from eukaryotes. That is a hypothesis. Probably not. You would have to reverse a lot in evolution, right? But that is a possibility. The reason they're sister to each other is because we share more similarities in our DNA with archaeans, when I say we, eukaryotes, than bacteria do to archaeans. So archaeans and bacteria are pretty different, actually, right? But yeah, any tree, and that's a great, it's 3-dimensional, so you can flip the groups. They can rotate. They're still on the same branch, right? If you rotate them. So yeah, this is not a great tree. They should have drew it so that it, I understand what you're saying. Yeah. Okay, so let's talk about bacteria first. So bacteria are the oldest group. They are the most diverse group, and they're the most abundant group. And it makes a lot of sense because they were the first ones here, right? First ones to occupy the planet, first ones to fill all the niches. And because they're simple, they can divide very quickly, and they have very diverse metabolic and genetic lineages because of horizontal gene transfer. So there are most likely more than 1 million or even 100 million, or some people say billions of species of bacteria. I don't know what the right number is, right? There's a ton of them. Now we have, as I've already mentioned, a really hard time determining individual species of bacteria because they don't fit the species definition, as we already mentioned. Because they're 100% asexual, they do come in 3 shapes, 3 major shapes. Circular, as you can see here. This is what is called coccus. So coccus is circular, C circular. They can come in a cylinder or elongated form that's straight, that's called bacillus. And then they can come in a spiral or spirillum form that looks kind of like a very small worm, right? And these are often called spirochetes. So these are the 3 cell shapes you can take if you're a bacterium. Most species that are probably, and the most species that we know of, are harmless, which is really important for me to make sure you guys understand, is that most bacteria do not harm us at all. And most of us, what's the first thing you think of when you think of bacteria? Icky, infection, right? Pathogen, plague, right? Death. So, and that's true. Bacteria are, many of them are harmful. But most bacteria are very beneficial to the planet and to you. And they're mutualists. That is, they live alongside you, often helping you. Or they're at least commensalist, and they don't harm you at all. They're just occupying a niche or space. Your body is covered in bacteria, inside and out. You're a superorganism. You have lots of bacteria diversity and species living on you. There are more bacteria in your large intestine than the number of cells that make up your body. All right, so they are a major part of our biology. Can't live without them. They cover everything. They're all over the surface of everything. You've grown them, right? This is just the surface of the human body and the different diversities of bacteria that reside there. Your body is an ecosystem to them. Their biome, if you will. And so some of them live in very specific places. The axilla, the bottom of the feet, between the toes, on the face, on the arms. There's completely different bacteria diversity that lives in different places on the body. They're in your nose, they're in your gut, they're all over the place. So you are a superorganism, as most vertebrates are. There are a lot of bacteria that help you survive from day to day. Now, some of these, or if some of them are removed, then pathogenic bacteria can take their place, right? And that's when you start getting illness and infection and whatnot. So bacteria abundance and metabolic diversity. So Earth's most abundant organism are the bacteria. This is the number of bacteria, nonillion, 5 in nonillion. Do you know what that number means? Neither did I the first time I saw the number. It's 1 with 30 to 50 zeros behind it, depending on whether you're in the US or you're British. It's a huge number, right? That's how many, you can't even fathom it. That's how many bacteria are sitting on the planet alive at any one time. But again, most abundant and most diverse, but we know pretty much nothing about them. Which ones do you know about? You know about the ones that are pathogens, right? Those are the ones we understand the best. Those are the ones that have been studied the best, or the ones we can grow on petri dishes, right? Those are the ones we understand the best because we can actually do some actual scientific experimentation on them. There are only 9,000 or more, closer to 10,000 probably now, species of bacteria that have been described. Now, how can we break bacteria down? So if we look at bacteria, our first domain, there are 5, we'll just say this is how your textbook puts them together, and this is pretty universally accepted. There are 5 major phyla of bacteria. Sometimes they're called superphyla. So these are proteobacteria, chlamydia, spirochetes, cyanobacteria, and then a really large group they call gram-positive bacteria. So gram-positive bacteria are different than these other 4 groups, which are called gram-negative bacteria, based on the way they stain. All right, so the way they stain. That is, when I add a stain to them on a slide, some of them are purple and some of them are pink. If you're pink colored, then you are gram-negative. If you're purple colored, then you're gram-positive. Right? And so you might wonder, well, why, right? And 2 slides down from this is this picture, right? Process of gram staining. So we're going to use 2 stains. We use a pink stain and a purple stain. The big difference here is the purple stain binds to the cell wall of bacteria. And so a gram-positive bacteria has a very thick cell wall. As you can see here, here's the gram-positive. It has a very thick cell wall on the outside of its cell membrane. This wall is made up of a specialized polymer that's called peptidoglycan. The peptido means protein, glycan means sugar. This is a polysaccharide, a really long sugar with amino acids mixed in. And it's very specific to prokaryotes. We don't have anything like it really that make up the cell wall. The cell wall of gram-positive have many layers of this peptidoglycan polymer that binds to the purple stain. And so when you stain with the pink and purple, the purple is darker and binds to this thick wall, and these gram-positive bacteria look really purple, really dark colored. If you're gram-negative, you have 1 or 2 layers of peptidoglycan surrounded by lipids and sugars and your cell membrane. You don't pick up the purple stain at all, really, and you only pick up the pink stain. And that's why you look pink instead of purple. And so all gram-negative bacteria have a thin cell wall. All gram-positive bacteria have a very thick cell wall. Pretty simple. And so we can group them arbitrarily by positive and negative gram staining. All right, so these are gram-negative. This is gram-positive. Probably the biggest and most diverse group of bacteria are the proteobacteria. So you can see them up here. Here's the proteobacteria. And there are like 4 major groups or taxa under this particular phylum of bacterium we're going to call proteobacteria. So largest diverse clade of gram-negative, and they include a lot of different type of metabolic diversity as far as how they obtain and use nutrients. And we're going to go over these next time, but they have photoautotrophic, chemoautotrophic, and heterotrophic examples. When you see autotrophs, that means they make their own food, right? Heterotrophs have to ingest their food. So a photoautotroph is an organism that uses light, right? It does photosynthesis. A chemoautotroph uses chemicals, or it kind of takes up compounds that are not often carbon-based and use them to make energy. Great example of this is there's one chemoautotroph, actually there are a couple of these in this group, that are sulfur bacteria. And they use hydrogen sulfide, they oxidize it, they remove the hydrogens off of it, and then make energy, or ATP, by using an electron transport chain to pass those hydrogens around. Right? And so this is a chemoautotroph because it's not using sunlight, it's not using a carbon source, it's using this gas, inorganic gas, to actually make energy. Weird, right? It's something that most organisms that are alive can't do that. Others are simple heterotrophs. The heterotrophs are often pathogenic. They have to eat or obtain their nutrients. And the pathogens that are found in proteobacteria are things like gonorrhea, cholera, right? Have you heard cholera here? You ever heard of cholera before? It's a bacteria infection that causes mass diarrhea. It's from contaminated water. It can kill you if you don't have antibiotics. Before the antibiotic age, cholera killed lots of people. And then helicobacteria, a bacteria here, are those bacteria that cause bad stomach ulcers. So often if you have the symptoms of ulcers, stomach ulcers, they can't get rid of them, they will want to go in and take a sample of stomach to see if they can find this bacterium, and then they can treat it with antibiotics. So those are the proteobacteria. Again, most of them, again, are commensalist or mutualistic. They really don't harm humans. But there are some pathogens in there. Chlamydias are parasitic bacteria. They're really small, and they're parasites of animal cells. And so this down here is I know it's a small image, but this is a cell right here, an animal cell. And chlamydia bacteria, the chlamydiids are very small. So this little circle here, this is one of them. Here's another one. You can see that they have to get into the cell. They're intracellular parasites in order to survive, to get their nutrients and to divide. And I know many of you have heard of chlamydia. Chlamydia is the most common sexually transmitted disease in the US and almost worldwide anymore. And many people, a majority of people that have chlamydia don't have symptoms, so they have no idea they have it. They have to be tested for it. But it's passed through sexual contact, can pass through swapping of saliva, using makeup. It can be passed pretty easily. If it gets into the eyes, either passed by body fluids or from touching genitalia and touching the eyes without hand washing, it can cause blindness. It can attack the corneum and cause blindness if it's not treated. That's why it needs to be treated with antibiotics. And it is the leading cause of blindness across the world. Pretty interesting. So those are the chlamydias. So they're intracellular parasites. The spirochetes are these spirillum-shaped bacterium. They're almost all heterotrophs. So they get their nutrients from the environment. Some of them have flagella. Some of them just wiggle or rotate. Many spirochetes are free-living and not pathogenic at all, but there are several pathogenic parasites here. The two major ones that you've heard of before are syphilis, right? which again was very deadly until antibiotics, and Lyme's disease, one that's really prevalent here in the Midwest now, particularly in Indiana.
Do you get that from mosquitoes?
Get it from ticks, deer tick, yeah. So this is a deer tick here in the Midwest and the East, and then there's a western tick that also carries it, but it's more prevalent here in the Midwest than the northeastern United States. And again, a lot of people that have Lyme disease never have symptoms of it, but others have dramatic symptoms from it depending on the way your body reacts to it. It also can sit dormant for a long period of time in the body and then all of a sudden affect the nervous system and other body organs. Cyanobacteria are photosynthetic, they're autotrophs. They are marine and freshwater. And these guys are, their ancestors of cyanobacteria are probably the ones that became plastids in the first eukaryotes and allowed them, the first eukaryotes, to do photosynthesis. They were also, their ancestors were the first to add oxygen to the atmosphere and kill all of the more ancestral anaerobic prokaryotes that were around the planet before oxygen was made available by the cyanobacteria. These guys can be solitary, or often you see them in long filamentous strings of oily, very green masses that sit on the top of stagnant water. Have you ever seen that before? It's got like an oil slick, all this nasty green material that's sitting on top of stagnant pond or lake. Those are cyanobacteria. That long strand that's kind of plant-like is not a plant. It's actually millions of individual cyanobacteria glued together by a mucus-like protein. And they can produce a lot of cyanobacteria toxins that you don't want to drink that water, right? It can cause a lot of problems in the human body, or can cause dogs problems, for example. They drink nasty cyanobacteria-rich waters. In the marine environment, these are major parts of the phytoplankton and are major parts of primary production and produce a lot of sugars using sunlight, and are a major part of the base of the food web, if you will, in the marine environment. The last group are the gram-positive bacteria. These are the ones with the thick cell walls. Again, many of these are soil-dwelling and mutualistic and commensalists, but there are several of them that are the more famous bacteria, like Staphylococcus and Streptococcus. These are gram-positive bacteria that are cockle in shape. Staph, particularly Staphylococcus aureus, is what forms all the little infections you might get if you chew your fingernails, for example, or the pustules like acne are staph infections. Staph infections, if they go crazy, can be, pretty detrimental to you. So again, because of antibiotics, we can treat staph infections. Anthrax is in this group too. Bacillus anthrax, which anthrax affects the lungs. Clostridium botulinum causes botulism, which is when you get canned goods that get dented or opened and the bacteria gets in, and they divide in that environment until they divide so much that they use all the oxygen and kill themselves. And when they die, they break open and release toxins into the soup can that can be deadly. All right, so it's really rare, but that's why you don't buy dented, soup cans, for example. So those are the gram-positive bacteria, and so those are the major kind of groups of bacteria that we have, and this is the phylogenetic tree of them. What do you notice about this phylogenetic tree that's different than all of the others that I've showed you? It doesn't have a what? It's not rooted, right? It doesn't start off as a line down here and then all the groups come off, right? It's like a spiral around a central point, because we don't know the common ancestor, right? We don't have Luca. Luca would be our common ancestor. When you don't have a root to your tree or a common ancestor to your tree, you have this circular pattern. And I mentioned during that lecture that you have all these words up here that end in troph that are modes of nutrition. And so that's where we're going to start today. And therefore, once we do that slide, you can go back and notice that, proteobacteria, one of the reasons they're so diverse is because they have lots of ability, if you will, to deal with nutrition, right? And so they can be photoautotrophs, chemoautotrophs, and heterotrophs. All of those are different modes of obtaining nutrients. Whereas most cyanobacteria are photoautotrophs. They are photosynthetic, right? And are a prime example, they're ancestors of where chloroplasts came from, probably, right? With endosymbiosis. So these are the nutritional modes, this figure here, or table I should say, sorry, that comes out of the Campbell textbook. Shows you the major modes of nutrition for prokaryotes. And a lot of these you can apply also to eukaryotes. So really the name has basically a first part, a second part, and then troph at the end. Troph or trophic just means mode here or type. The first part of the word, photo or chemo, simply tells you where you're getting energy to produce sugars or to make ATP. The second part of the word, auto or hetero, simply means where you're getting your carbon from. Typically, autotrophs use carbon dioxide to make sugar, right? So what are you doing to the sugar is you're adding electrons and hydrogens to it, right? We call that reduction, right? When you reduce something, you add hydrogens and electrons to it. When you reduce something, when you add electrons and hydrogens to something, that energizes it or adds energy. It's a way you store energy. And so you'll go from carbon dioxide to glucose, which is C6H12O6, right? You added 12 hydrogens to it. To do that, to add energy and electrons and hydrogens to a carbon source, you need an energy source. So photoautotrophs use light, right? So they're using sunlight to split water in photosynthesis, gather the electrons and hydrogens, and add them to carbon dioxide. The byproduct of that, or the release of that, is oxygen, right? You release oxygen from that process. Now, if you go down one, chemoautotrophs. So again, we're using carbon dioxide, but we're not using light anymore, right? We're using chemo, we're using inorganic substances to actually gather energy. So we're going to break bonds and capture that energy from the broken bond, and then use that to reduce my carbon dioxide into sugar. And so that's what chemoautotrophs do. It's actually pretty cool. They don't need light. Often these guys, the chemoautotrophic prokaryotes, are what are called obligate anaerobes. All right, they're called obligate anaerobes because they hate oxygen. Obligate means that they have to have a certain condition or they can't survive. They're obligated to be anaerobes. So if oxygen comes into their environment, they die, all right, because they are anaerobic. So an obligate, wow, guess I have my computer. An obligate anaerobe is going to be one, stop doing that, that cannot survive in environments with oxygen. An obligate aerobe, or an obligate aerobic prokaryote, has to have oxygen in order to survive. It's like us. If I put you in a no oxygen environment, I throw you into space, you're going to die, right? Because you need oxygen to make energy. And so there are also prokaryotes that have to have oxygen in order to survive. So those are called obligate anaerobes and obligate aerobes. Facultative anaerobes here are flexible. That's how I remember it. Facultative, F, flexible. They don't care if there's oxygen. They can live without oxygen, so they can survive in both environments. They're a little more adaptable, if you will, as far as oxygen goes. The last two here are heterotrophs, which means instead of using carbon dioxide for their carbon source, they're going to ingest more complicated compounds, more organic compounds. So things like small sugars or alcohols, they can ingest, and then photoheterotrophs will ingest small organic compounds and use light to make more complicated sugars that they can then use for energy. And the last one is chemoheterotrophs, which take in organic compounds and then use those to make energy. It's like us. We eat to make energy, right? And to get the building blocks that we need to maintain our metabolism. So plants are photoautotrophs. There are algae and protists that are photoautotrophs. And we, animals, and many protists, as we'll learn when we get to eukaryotes, are also chemoheterotrophs. Right? We have to ingest our food in order to obtain energy. So when you see these words, or you're learning, that proteobacteria are photoautotroph, chemoautotrophs, and chemoheterotrophs, then you'll know what those terms mean when you can come back and look at this table. So that's bacteria. Now we're going to do archaeans. So basically, we talked about Luca yesterday. We did the bacteria line, the oldest living line of organism. And now the archaeans, which are more closely related to us than bacteria, are in a really neat group. They have all of the prokaryotic characteristics. They're unicellular, they're 100% asexual, they have cell walls, they have a circular chromosome, they have plasmids, so they look a lot like bacteria. But they have a lot of characteristics they also share with us, or that are similar to eukaryotes when I say us. And I'll give you a couple of examples. Number one, they have histone proteins. All right, histone proteins, remember, are the proteins associated with DNA, right? Their histones are very similar to ours. All right, so bacteria don't have histones similar to ours. They use methionine, which is the first amino acid in every protein that a eukaryote produces during translation. They use methionine the same way we do. What I mean by that is every protein starts with methionine, right? It'll be the first amino acids that's brought in. Archaeans do that. Bacteria don't do that. They have very similar transcription and translational metabolic pathways to us, and multiple types of polymerase. All right, so polymerase is the enzymes that are responsible for making RNA and DNA. So they have multiple types like we do, whereas bacteria do not. So they share a lot of these metabolic and molecular types of characteristics with us that involve DNA and metabolism. So that's why they are sister to us and separate from bacteria. Now, almost all archaeans, and at least the ones that we really know well, are called extremophiles. So phile means to love, extreme means environments. So they typically can live in environments, many of them can, that no other organism can, like the hot springs that are way above a temperature we could tolerate. It would boil us. Right? So they can live in hot springs. So all this beautiful color that you see are mats of archaeans that are photosynthetic, because some of them are photosynthetic. Most of them, and a lot of them also can be heterotrophs, but they can live in pure salt, for example, is another extreme environment that they can live in. So Salt Lake, the Great Salt Lake, the Dead Sea. If you look along the shore, there'll be some beautifully colored stripes and striations in the pure saline solutions there that are archaeans. They can live on the surface of ice. They can live, and as I mentioned, at high temperature. So they can live in all these really extreme places because of their protein structure and their metabolism. The other pretty cool thing about them are there are no known pathogens that are archaeans. So none of them cause infection in us like bacteria, some bacteria do. They split supposedly from the bacteria line if you look at their molecular clocks about 3.8 billion years ago. They are an old line, but they again share a lot of characteristics with us as far as metabolism and DNA. Species richness, like any prokaryote, is not known because of our lack of technology to separate out species, like we talked about yesterday. All right, so that's all I'm going to spend on prokaryotes. Now we're at the rest of the slideshow, 60 slides or so, is on the eukaryotic line, or eukarya. So we got those three domains, right? Bacteria, archaea, and eukarya. And you can see behind the word here that we got all kinds of different types of eukaryotes. These are the organisms you're probably most familiar with, right? We're going to move into the animals and start talking about animals as we go through our taxonomic review, if you will, of eukaryotes. So we have described somewhere around 2,100,000 or so, give or take 50,000, or so eukaryotes. This number is probably closer to probably 2,300,000, 2,400,000 actually nowadays, but because we're finding new species all the time. But we still put them into what we call 4 clades. These clades are called supergroups. We call them superclades. That's what I call them, at least. They're these huge groups. Classically, eukaryotes are broken down into 4 kingdoms. Right? They're broken down into 4 kingdoms. That's how I learned them. Kingdom Animalia, animals. Kingdom Plantae, plants. Kingdom Fungi, fungus. And then protists, protista. You'll see that on another slide. Those are the 4 kingdoms. Those are all non-monophyletic groups. They're all unnatural clades now. And so we put them in these supergroups that are kind of between domain and kingdom. The 4 kingdoms are going to be nested, the classical kingdoms, within these 4 supergroups. Now, here's our little very simplistic tree, right? Here's Leuca, here are bacteria, the oldest group. Here are archaeans, sister to eukaryotes. So what makes eukaryotes different than the prokaryotes? These would be synapomorphies, most of these, right? For the eukaryotic line, which would separate them from Leuca, bacteria, and archaeans. So we've already heard of these, right? So membrane-bound organelles, linear, not circular chromosomes. Chromosomes meaning multiple chromosomes. Those chromosomes are housed in a big organelle called the nucleus. Some have cell walls, which is similar to prokaryotes, but the cell wall is completely different as far as what it's made up of. So it's not made of peptidoglycan, it's made of chitin or cellulose, as you'll see. Some are unicellular, again, a lot like prokaryotes, but there are a lot of multicellular examples of eukaryotes. And these multicellular, larger eukaryotes are billions of cells, like you, right? And they typically are much larger in size, each cell, than the prokaryotes. So here are your 4 supergroups. So here is a phylogenetic tree, again, a pretty simple phylogenetic tree, of the major superclades, if you will, of the eukaryote. So here is your ancestral eukaryote, which has all those traits we just talked about. And then you got these 4 groups: Excavata, the sarclade, archaeoplastid are archaeoplastids, and then Amorphia, right, at the bottom here, are the amorphids. So this first group is our basal group. So we're going to start up here and then talk about these guys and then work our way down to animals. That's really what we're going to do in the rest of the PowerPoint. And so you got your 4 supergroups. Now when you look at these, everything in yellow is a protist. So that used to be a kingdom, right? We used to call it Kingdom Protista. But notice it's obviously really polyphyletic, isn't it? They're all over the place. You know, you can't put them into one monophyletic group because, look, they all share different recent common ancestors, right? Of course, yes, you can trace them all the way back to the original, but, you know, for this to be just protist, we would have to forget those, and those, right? They all share different common ancestors. There's your kingdom plantae, right? Your land plants. There's your fungi, kingdom fungi, and there's your animalia. So your kingdoms are nested within the supergroups. So let's talk about protists first. So protists are a huge group. Really, back in the day, I used to call, don't tell Dr. Sweets this, I used to call them a junk group. Because if you weren't a plant, an animal, or a fungus, you got thrown into the protist, right? And so they have tremendous diversity. There's over 200,000 species. You know, that's a conservative number. There's probably many more than that that have been described. And the thing is, if they're polyphyletic and share all these different common ancestors, there's no real one single group of traits that's unique just to protists. So they're really hard to define. So we do things like how many cells do they have? What do they eat? How do they reproduce? What's their ploidy? Are they haploid? Are they diploid? Are they polyploid? We use those types of characteristics to group them together to study them. Those groupings based on these characteristics are not necessarily phylogenetically sound, right? Now this picture right here, this is an SEM enlarged of the exoskeleton or the home that a diatom lives in, right? And so this is what's left over when the diatom dies. And it's a silica casing, if you will, that is heavy and it'll sink when the animal dies. So here are some cool examples, or interesting examples of protists. So remember, we saw a lot of these in the pond water in the beginning of the class, right, when we did microscopes. And so that's the fun thing about pond water is you never know what you're going to see. It's like Christmas, right? You never know what animal you're going to see. Now, many of these are tremendously bizarre. Like, look at this one. So this is a rhizarian. We're going to talk about what a rhizarian is in a minute. It has this is just the exoskeleton. It's just, they're really bizarre and alien-like, and they're really, really tiny. They come in all kinds of shapes and sizes. Most of them are free living. They're out there living their life. They're not bothering us. But there are several of them that are very pathogenic and devastating to the human population, like this one, right? So that's a picture of this sporozoite of malaria. Malaria has killed more humans than any other catastrophe disease you can think of. Matter of fact, there are some scientists that estimate that half humans that have ever lived have died of malaria. So that's a lot of people, right? So we're going to talk about malaria a little bit when we get to that group. I have a little video here that, well, doesn't have any words. It'll just introduce you to protists. If you forget, what you saw in the pond water, I'll give you a little video of that so you can see it. So this is a cool video. I'm very jealous of this guy's microscope. Oh, the sound's on. Yeah. Very dramatic. Turn it down a little bit. Oh, you all want to be protist biologists now, right?
Are they still multicellular?
Hmm?
Are they still multicellular?
Are they mostly multicellular?
Yeah.
And well, most of them are unicellular. I mean, there are some multicellular ones for sure, but you have unicellular, multicellular, and even colonial, which aren't true multicellular organisms that are protists. So they kind of defy, one form or another. There are great examples of all three of those kind of organizations. They have all the eukaryotic characteristics. Yep. So they have organelles and nuclei and all of the same characteristics that we do. Matter of fact, they have a lot more, since a lot of them are unicellular, they have a lot more specialized organelles that we don't have, like contractile vacuoles and a number of other types of plastids and structures that you don't see in a typical like animal, for example. Okay, this one is just an embedded video here. This is a pretty cool video that shows you vacuole formation in the typical ciliate here, or what we're going to call paramecium, which are in excavata. And if you look really closely, these guys are called excavata because of the long canal here that's called the food groove or the oral groove. And you can see there's a membrane that's on the edge of here, and it swells into a vacuole. As you see, it collects food, and then it pinches off and forms these cool food vacuoles. So they collect the food through cilia. They basically are sucking in through this groove food and then emptying it into food vacuoles, and then the food vacuoles enter the cytoplasm. They merge with a lysosome, and that's how they digest food. So they're heterotrophs, paramecium are, chemoheterotrophs like us. So let's talk about the first group. All right, so this is a little phylogenetic tree down here that just is showing you the top of the phylogenetic tree, the first group here. These groups down here would be the sarclade. Up here, on an open phylogenetic tree here, we can see the excavates here, excavata. Looks like and sounds like the word excavate, right? And so think about a little excavator that's making a long canal in your front lawn so that you can bury a pipe, right? If you look down on that canal, that's what their feeding groove looks like. It looks like somebody excavated out a feeding groove. And that's where the name comes from, like I just showed you. And then they can suck in food from the environment and make food vacuoles from those. Some of them have cilia, some of them have flagella, right, like you can see here. Most of them are free living, all right. Whoops, went too far. So most of them are free living, but the first two groups, the diplomonads and the parabacillids, these are parasitic groups, and there are a number of different types of infections that they cause, but the two major ones are the two common ones that you may have heard of. The first one's called Giardia. You guys heard of Giardia before? That's why you don't drink out of a creek or drink directly out of a pond, because this is a common genus or group. Giardia is a genus that's found in the diplomonads that cause digestive problems, diarrhea, bloating, destruction of the lining of the small intestine that can lead to complications over the entire digestive tract if not treated. Giardia is treated with specialized antibiotics if you were to ever get it. The other group down here is called the parabacillids. They have a number of genera. The genre of interest here is Trichomonas, which forms trichomoniasis, which is one of the more common sexually transmitted disease that's protozoan in nature. So you can also treat it with antibiotics. The other large group here, the third one, are called euglenozoans. And these are mostly, not all of them, but mostly free living, not bothering us. There are a couple of pretty nasty, actually, pathogens in the group. And we'll talk about this group in a little more detail. So very diverse, the euglenozoans. Mostly heterotrophic, but there are autotrophs in here, photoautotrophs. There are actually a number of euglenozoans which are called mixotrophs. So a mixotroph contains 2 or more nutritional modes. It can use 2 or more nutritional modes. Euglena is a great example of this. So here's kind of a still image of euglena, and then this is, I think, is also a species of euglena. They are heterotrophs. They can go around and eat stuff, no problem. But they also have chloroplasts, so they can make their own sugars. So when there is no sunlight, they eat. They're heterotrophs. When there is lots of sunlight, then they are photoautotrophs. So they can kind of do both, which, man, what a great adaptation, right? To be able to do both. Now there are a couple of pathogenic parasites in this group. Probably the one that's most famous is African sleeping sickness. Here's the genus of protist that is responsible for African sleeping sickness. It's called Trypanosoma. This this particular parasite, like most parasites, has a very complicated life cycle. There are 2 hosts. The first host is called the tsetse fly. So it's an African fly group that bites humans. It feeds on blood. And if an infected fly bites you, it will basically inject into your blood the trypanosome. And this is what a trypanosome looks like. This is the motile stage traveling through the blood supply. That's an SEM of the little guy. These guys move around and invade organs, and eventually, if they're not treated, they can move into the central nervous system and cause African sleeping sickness, where you fall asleep, it's hard to wake you up, you fall into a coma, and you die if it's not treated. So it's a pretty nasty bug. Now, all euglenozoans, I shouldn't say all, the majority of euglenozoans have a crystalline lattice work like this. You can see all this kind of lattice work or laid down material that is in a very organized fashion. That's called the crystalline rod, that is associated with one of their flagella if they are flagellated. Many of the euglenozoans have two flagella. One of them, or both of them in some cases, have this rod associated with the microtubule. So if you look right here, this is your 9 pairs of microtubules in your central doublet. This is the microtubule arrangement of the flagellum coming out at you, right? It's been cut through a transmission electron microscope image, and the crystalline rod that sits next to it. We have no idea what it's for. We assume structural support. But it's a diagnostic character of a euglenozoan. If they have that and you're a protist, you're probably a euglenozoan. All right, so those are the excavates. So here's a little, video of one of these guys that obviously is photosynthetic with the green pigments, right? It's got lots of chloroplasts. Okay, the next clade is the clade just underneath Excavata. So you can see here is my phylogenetic tree, and I've closed down Excavata. I have the Archaeoplastida down here. And you see this word uniconta. That is an old school term for amorpha or amorphia. So we've opened up the SAR clade, and you can see there are a bunch of them, right? And these are all protists. Bunches of groups in here, so I'm not going to make you learn all of them, right? I'll save that for biodiversity class. And because have a time restraint, right? But here are your 3 groups that make up SAR. So the word SAR, or this acronym, comes from the S, the A, and the R, and the 3 major groups that make up this clade, this supergroup. They're called Straminopiles, Aviolates, and Rhizarians. All right, are the 3 groups, and I'm not going to teach you all of them. I am going to, for your example, of strominopyle, we're going to look at diatoms. For your alveolate, we're going to look at the apicomplexans. And then for the rhizarians, we're not really going to talk much about them. The only thing you need to know for the rhizarians is that they have a exoskeleton, or an outer kind of a house, if you will, that they produce. And it is calcium carbonate based. They're marine. And these two pictures up here are pictures of rhizarians. I'm not going to teach you any specific examples of them, but they do produce what look like little seashells. All right, cute little seashells that are microscopic. Like this one looks like, like a mollusk, right? But it's not. It is just a calcium carbonate exoskeleton that was laid down by the rhizarian that is now dead. Here's another one with the rhizarian exoskeleton that you can see it's calcium carbonate based, and they send out little pseudopodia, or arms, right? So these pseudopodia so this word pseudopodia, pseudo means false, podia means feet. They're just elongated extensions of the cell membrane and cytoplasm. Remember microvilli? Now just think of really long microvilli. So they're really elongated so they can stick out the holes. And almost all the rhizarians are heterotrophic, so they capture stuff with those little pseudopodia, and they hide in these little tests. So, and you can find the little test in the sediment. After they die, they settle down, and they're actually kind of cool looking under a microscope. That's really all you need to know about the rhizarians. I'm going to give you your two examples here of the other two, the strominopyles, diatoms, and the alveolates, malaria. So the words here, strominopyle means straw hair. Actually, that's the literal Greek translation. Straw, like a long, hollow straw, hair, which stands for their really long pseudopodia. So they have very, very long pseudopodia. And I'm going to move to the next picture, that they use to stick out of their little homes that they produce, these exoskeletons. Their exoskeleton is made of silica. It's glass. It's a lot like glass. It's glass-like. It's very clear. So here are some diatoms. Many diatoms, or almost all diatoms, are photosynthetic. So they're photoautotrophs. They have chloroplasts. And they use these little pseudopodia to move around and interact with their environment. So you can see them kind of glide across surfaces. Like you can see here, this guy's gliding. You can see the really elongated, hollow straw-like blue stringy material sticking out of this silica exoskeleton that they use to move around. These guys are a major component of phytoplankton in marine and freshwater environments. So they are a major contributor in aquatic environments to a term we used yesterday called primary production, right? So they are the primary capturers of energy into our system here, light, to use photosynthesis to produce sugars. And they are the base of the food chains in almost all aquatic environments. There are so many of these across the history of the earth that as they die, they settle to the bottom of ponds and the oceans in particular and form sediment that's called diatomaceous earth. It's diatoms basically piled up on top of one another that have died. And they make up the major part of what we call the plankton layer, which is the layer that's on the top of the ocean or a lake where sunlight hits. Because they're a major component of primary production, and if you imagine in the marine environment, if I tell you they are one of the major primary producers in all of the oceans of the world, then they are using a ton of sunlight and a ton of carbon dioxide, right? They're using lots and lots of carbon dioxide over the entire surface of the ocean. Because of that, their photosynthesis helps determine our climate. All right, and so you might wonder how is that possible? Because remember, primary production uses carbon dioxide, right? Uses carbon dioxide and light to make sugar. So we're using carbon dioxide and light to make sugar. Yesterday we talked a little bit about Earth's history. Remember snowball Earth? Because of the oxygen revolution and the removal of carbon dioxide from the atmosphere. Carbon dioxide forms an insulative blanket around our planet that traps heat. And as you know, and as you've heard of, we're undergoing right now, as humans, the production of more carbon dioxide than is produced naturally, right? And we're adding to this what we call greenhouse effect, right? The planet's warming. And it is warming. You can't argue with it. Even if it snows like 5,000 inches this winter, which I doubt it will, that doesn't mean that we're cooling down, right? That's called weather. I'm talking about climate, long-term climate. The Earth is warming slowly from year to year, if you look at overall temperature across the planet. And that's because our carbon dioxide layer is increasing as we add more and more carbon dioxide to the atmosphere from burning fossil fuels. These guys, as long as they're really high in number in their aquatic environments, they remove that carbon dioxide to make sugar and release oxygen. So they're preventing our planet from warming up too much. Right? They don't remove all the carbon dioxide, but they prevent it from getting too thick so that our planet doesn't warm above what life can tolerate. Does that make sense? The other cool thing about them is because they have a silica skeleton that's kind of heavy, when they die, they take that carbon with them. They drop to the bottom of the ocean and take all that carbon dioxide that they had taken in to the bottom of the ocean with them. And they die, and all of that carbon goes into the soil. And so they are called, because of that, they are called a carbon sink. Right? In the sense that they trap carbon dioxide, take it out of the atmosphere and trap it, and then drop to the bottom of the ocean where it's stored in the sediments. Again, preventing carbon dioxide from building up around the planet itself, in the stratosphere, around in the atmosphere. So they're very important ecologically. So that's your diatom, your stromatopile, that example. Our last example we're going to look at least today, and we're probably not going to get through it, are the alveolates. Now the name alveolate refers to a structure called an alveolus, which if you've taken any anatomy is in our lungs, right? It means air space, all right, or air cavity. And so these guys are named because underneath portions of their cell membrane, they have these big vesicles that look like little alveoli or air spaces under a microscope. And that's where the name comes from. All these are fluid-filled sacs that increase surface area and allow them to perform certain functions for the protist. In the apicomplexans, they have taken these alveoli and formed a specialized structure at the apex of the cell, or at the top of the cell. So here is one of the stages of the parasite that we call malaria. It's called the merozoite, which has this little conical structure on its apex. And this structure that's in black is the modified group of alveoli that are responsible for allowing the merozoite to attach or suction cup itself to the surface of our red blood cells. And then what it will do is release a modal mechanism that will part the red blood cell membrane so that it can enter the red blood cell. So it's got to attach and then enter the red blood cell using the modified alveoli. And that's why they're called apicomplexans, which means a complex apical or pointed end. Once they get into the red blood cell, they actually stain like a little ring. So here are the merozoites inside of the red blood cells. So we know from this blood smear that's stained specifically for the merozyte that it actually is, this person has malaria, right? And so they hide inside of the red blood cells and mature inside the red blood cells. So apex is complex. The alveoli have been modified and specialized for parasitism of our red blood cells, helps them be very effective parasites. Some cause serious human disease like malaria. Malaria does not only attack humans, it does attack other mammals. Malaria does have a very complicated life cycle, which we're going to learn. We're going to come back to life cycles. Woohoo! Learn their life cycles. There are two hosts. We're one, obviously. And then mosquitoes, right, are the other one. Mosquitoes are the other part of the life cycle. The protozoan that infects humans is called Plasmodium, all right, falciparum. Plasmodium is a terrible organism as far as human infection. And as I mentioned before, it has been estimated that somewhere near a half of all humans that have ever lived have died from malaria. So I'll show this video here